Hello, everyone. Welcome to the speaker panel. Thank you for staying. Before we continue, we want to take a moment to thank everyone for your attendance and being here all day. It's a long day, but it's an exciting one. And thank you again to our speakers for taking the stage once again. Just when you thought the anxiety was over, we're back. The goal of this panel is to bridge topics across all the disciplines that we've covered today, interconnect themes from the day, and understand further the technologies that can advance plant sciences. During the panel, we will be taking questions from the audience or through Slido and circle back to questions that could not be answered earlier in the day. We'd love for the panel to be conversational. For this reason, we encourage the panelists to respond to each other and contribute each other's responses. You can use the Slido QR code on your tables to submit questions, which John and I will read, or simply raise your hand and a PSGSC board member will come by with a microphone. So to get us started, John will take over.
Yeah, so while we kind of give you guys a few minutes here to formulate your questions and get them submitted or find a mic, I'm going to get us started off with, and as we have talked throughout the day, our theme has been growing into new tech. How are new emerging technologies impacting the plant sciences broadly? And the question that I had canned for each one of you today is, what technology in your field are you most excited about as possibly being the next paradigm shifting technology?
So I think on the specifically with regard to forensic wood identification, what it's going to be when we have a very large image data set collected with the Xylotron, and right now we're using deep learning to extract the features that are not explicitly anatomical or understood by humans. So we're working on explicit semantic segmentation so that we make real measurements of the actual cells themselves rather than just, textural features that the computer finds in the images. And when that happens, we will pretty much literally overnight have the world's largest quantitative data set for wood structure that exists. And that'll be for hydraulic architecture, that'll be for wood identification, that'll be for quantitative wood anatomy, and once our segmentation tools hit, it's all done in one fell swoop.
This might be a little bit of a non-intuitive. I would say the thing that I'm really excited about is when we get satellite imagery at a higher resolution down to the plant level. I can take a sprayer. We were talking earlier. We can take a sprayer through the field, but we have to hit certain speed requirements, size requirements to be able to bring something to market. I still think there's a lot of value in drone imagery, satellite imagery. I prefer satellite, just personal preference, because I think it's easier to capture over time. I don't have to send somebody out there with a drone. And so I'm very excited to see satellite imagery get down to very, very high resolution and then start coupling that with some of the images we get off of our machines.
So following that, I actually have a question about the satellite images. The biggest concern with using the satellite image is going to be cloud cover. So it has long interval, usually 5 to 8 days or even longer than that. And then on the day, if let's say it's on top of your target field and is your cloud cover, then you're missing the whole thing, right? You have to wait until the next cycle. So how do you view that?
Very interesting question. I was actually talking to a farmer in Malaysia yesterday about this, and they're trying to do new crop management practices. And I was like, "Oh, so you guys are doing satellite imagery?" He's like, "No, we have too many clouds in Malaysia because of the high humidity." And so I think it's a combination of the technology that will help us take a step forward. In his case, he has pivot irrigation on his farm, and they're putting vision systems on the pivots, coupling that with drones, coupling that with satellites when they can get the imagery. And those layers together can paint the picture of the farm. I don't think we're ever going to be able to just have satellite imagery or just have imagery off of a sprayer or something like that to solve the problem. Because the same problem we have with a sprayer, right? If I only take my machine across the field once or maybe twice in the summertime, that's not enough data to be able to make informed decisions about the crops. So I really think it's putting it all together that's going to make a change.
Technology, it's great, isn't it? And I think I would kind of go, even take that to the point of where I get more excited is around taking all of these pieces, being able to put it together, learn from that, but ultimately, how do we make informed decisions early enough in the growing season that it's going to have that impact? You think about like right now, within Pioneer, we have a very large drone fleet, and we have our field agronomists are out flying. We have satellite imagery, so we have those capabilities. The challenge is it doesn't necessarily tell us. It tells us there was a problem, or it tells us that, say, there's a potentially a low yield area, but it doesn't tell us why. And I think that as we see these different methodologies coming together, if we can really start to understand the why, and then is there something potentially we can apply early enough in the season to affect that outcome, I think that's where I get really excited. And, I've seen these things from John Deere, and it's some pretty cool stuff. And, you think about, is that, while we don't ever want to replace boots on the ground, you always want to have that person out there. They can't cover every acre, but when you start seeing a camera that can look at every plant as you're going through the field, that's some pretty cool stuff right there.
So for everybody who's interested, there's look up a company called Interplant. They're looking at putting fluorescent genes into crops so that whenever you have a disease come, it's going to trigger a fluorescent gene. And they say that they can have the plants fluoresce with a signal that can be picked up by a drone or a satellite or a sprayer within 3 days of the disease first causing damage on the crop. That is early. That's when we can start making good decisions.
So actually, Matt and I were having a very exciting conversation during the break about these technologies and how to make early decisions based on the imagery or whatever. Can we tell as early as possible before the real thing happened? And to me, I think the most exciting technologies I have is the hyperspectral imagery, because the narrow bands that provide the continuous data of reflectance that can capture the nuance information in the crops. So we have some previous studies done for potato early blight and late blight disease management. They did find a correlation between the early signals in the reflectance data and then before the onset of the diseases. The really symptoms take place on the plants. So that will be my answer to the question, the hyperspectral. And then if we can integrate the hyperspectral imagery with thermal, because the change of the plant physiology will always go with the changes in the temperature, right? The respiration, the closure of stomata, and so forth. So I think using these two technologies together is going to help us to identify the early disease or early stress. And then Stephen and Matt are all mentioning the drone or satellite image. I think the ideal scenario will be that the satellite can carry really high resolution hyperspectral sensors. The thermal is going to be very costly, but if we don't consider the cost, hyperspectral and thermal ideally will be my preference, let's say 20 years from now. I know that NASA will launch a new satellite in the next 5-ish years with hyperspectral sensors. So hopefully there will be some publicly available data in the near future.
That was great organic conversation out of that. Thank you guys. We'll We have several questions. I know the first one was in the far back left corner, and we'll go there first.
Hi, I'm not sure if any of you have this expertise, but one thing I've always wondered about processing a lot of big data and having, I don't know, predictive, reasonable predictions about decisions we're going to make. Does anybody have any ideas of how that impacts decisions on crop insurance or other economic scenarios like that?
Steve Jobs always takes 20 seconds to think. Did you ever see that video? He takes 20 seconds to think before he asks. You know, a lot of what you're talking about comes down to risk management and thinking about how much risk is that farmer willing to take on, and then thinking from the insurance company standpoint, you know, the more risk, the more the challenges are going to be when it comes to the insurance aspect of it. And I think that, you know, one of the things that as we think about, you know, all this work around modeling is really how do we start to understand stability? You know, thinking about whether it's product development, how can we get products that work well across wide-ranging acres? And, you know, I think in the end, we're going to continue to see that, and we're going to continue to understand. And I think really, too, as we think about the farmers, we think about the insurance industry, it's really about how do we just start decreasing the risk of everything. So in the end, regardless if maybe we're in a dry year or an extremely wet year, maybe we have issues with our planting, maybe we can't get back in the field to make a second nitrogen application or what have you, that our variations in yield that we're going to see are less drastic as we go through time. So I think that's where we're starting to see that trend go.
Great. Let's do a Slido question just because a lot are coming in, and then we can jump back to audience as well. For Joe, how do you ensure that you have captured images of all the leaves just by taking top aerial photos for foliar disease screening?
We don't. Yeah, so we really, that's actually sort of one of the cruxes, I think, of that project is that you can only see what's visible to the UAV, which is flying above, and to make things a little more difficult, SLB at least has a tendency to start at the bottom and work its way up. That being said, if you just spend some time looking at the photos, there is pretty good penetration into the lower levels of the canopy, at least before leaves start senescing pretty badly. So, or to put it sort of another way, I worked with someone once who made the point that if you lay on your back in a plot of corn, you can see blue sky. Right. So the opposite is true. When you're flying a UAV, you do have a perspective of lower leaves. It's just not as complete as the upper canopy. So I'm optimistic.
Who among us has not laid on our back in our research plots and looked at the sky?
Yeah, out of exhaustion or desperation or.
Exactly. I know there were a few more questions out in the audience, so if we want to pass the mic to someone.
Thank you. It's fantastic to listen to you all about these technologies and their potential. My question is on the opposite side of that. We have a long relationship with these plants, multi-thousands of years connecting with these plant materials. And I'm wondering from your perspective, as we move towards imaging, the relationship with the plant materials changes. Our connection to the plant materials, for whether you're a farmer or whether you're a scientist trying to learn how to do biology, it's going to change. And I'm wondering if you all could speak to what you think the losses will be as we move into this very exciting technology. Thank you.
Sorry for asking such a depressing question.
maybe I can get started. I think it's not really the losses, but it will be a change of attitudes towards something new. How should I say this? I think I can convert your question to what are the biggest challenges for the long-term development of these technologies? To me, our industry folks might provide more insights, but to me, it's going to be the fear of farmers or humans to lose their power to AI or to the newer technologies. We all know that there's a shortage of labor in the agricultural or farming workplace. So we want to use the new technologies to replace tedious, repetitive, labor-intensive work. But the more refined tasks still need to be decided by the humans. But I think the biggest fear, the fear will be AI will replace my job. I'm going to lose my farm. Or I'm going to lose my power over my plants in the next 20 years. Not sure if I answered your question, Erwin, but that's my thought.
So I grew up walking fields when I was a kid. My dad's been an agronomist for 36 years. I've been around this for a really long time, relatively. And I think what we're going to lose is a connection to the social, personal impact that all the things we've talked about have on people. I remember very distinctly the first summer I was ever being a crop scout, scouting non-GMO corn in western Indiana, and that year there was a big influx of insects. There's a lot of corn borer coming that year, and I was scouting fields, walking fields, finding problems, writing them down, and I had to go drive up to the farmer gate and say, you have this problem, and I know this is going to cause you stress, headache, heartache, economic problems. You're going to have to make more passes than you want to. It's going to blow up your summer, and it's bad. I had so much heartburn over doing that, and I fear that gave me a lot of empathy for the farmers and the people that do this on a day-to-day basis. And when we look at disease and we look at these problems and we're imaging it, and great, we found it, and we're going to ping you and get a notification on your phone that you have this problem that you have to deal with as a farmer, I think it's very easy to lose the empathy for the real world impacts that the things that we're measuring have on people. And I am afraid of that, because it's not just data, it's people's lives, it's people's livelihoods. It's generations of family history that we have to be very sensitive about. As a field agronomist, a lot of what we do is, I'm a therapist, I'm a counselor. When things go bad, I tell people it's okay. Like, you got to still have that. And if we lose ourselves in the technology, we could lose our customers too.
I guess one piece I'd add to it, as we think about the ag industry as a whole, it's an industry based on relationships. It doesn't matter what part of it is. Now, at the same time, we think about as the generations change, do they still want those same levels of relationships? I think it is changing. Now, doesn't necessarily mean that we get rid of that. So I do see some potential issue of it becoming more of a transactional type of, there are transactions that happen, but there's a person, they're sitting down with you, they're making decisions, you're having that communication with each other. So I do think that as we continue to see that, could we start to see some of that loss happening? Possibly. Now, at the same time, I think that, where we do continue to have those personal relationships, and as we think about all of the technology that becomes available, does it help that person do their job better? I think so. I think we are going to get to that point also. So, yes, what we may lose in that personal relationship, but I think then we're going to be able to do those jobs better than what we had in the past. And personally, as an agronomist, I don't think AI is ever going to replace us as a whole. Maybe 10,000 years down the road. But,
Knock on wood.
Yeah, knock on wood. But you're still always going to need that. You're going to need that on the boots ground truthing of, is what we're seeing, is that really happening here?
Great, thank you. Let's do another Slido question. For Nate, can the image data be used in prediction of germination without actually conducting the germination test, and have you observed a correlation?
With the images that I showed, that the Z-stacked images that are featured, no. I mean, if they were really shriveled, we could maybe assume an aborted embryo. If that was the case, though, if the seeds were large enough to observe a shriveling effect, we'd probably be able to see that with our naked eye. On one of the first slides I had, there was like a viability testing slide. I had an X-ray image that we got from this newly acquired X-ray. With that, we can get, or somewhat, we can get an output that has a percentage of fill. Obviously, 100%, great. There's the huge presence of embryo. 0, there's no embryo. The 0%, for sure, that's not going to germinate. But the 100%, it doesn't mean that it'll necessarily germinate. And there may also be some dormancy complications in there as well. We also don't know at what percent for each species that partial fill would still have the potential to germinate. So we've got a lot of work to do to try to track that. We are working on it. We just acquired this piece of equipment in July. But we are measuring that and then taking the total from what we've sown for a viability test and comparing them. And we can highlight each one that was like partial fill. But it will take a long time, and for the time being, we would not use it in lieu of a germination test, and instead to couple them.
We've been getting a lot of questions coming in pertaining broadly to career advice, particularly for grad students, how you got to your position, what your pathway has been. So kind of covering that general area, if we could address several of these questions at once, that would be perfect.
That's a pretty broad question.
Yes.
30 seconds or less.
I agree it's broad, but I'm trying to make sure we get as many people's questions addressed.
Yeah. I'm happy to speak first. I don't have any like super insightful advice, except that reflecting on sort of my own path to where I am now, serendipitous things happen, and sort of taking advantage of those when they come along, or being able to recognize them when they come along can be really valuable. And of course, you never really notice those moments until hindsight. So good luck. And then also, I think the advice that we were sort of given earlier, which is recognize the things that you're good at and that you enjoy doing, and lean into them, and take everything else and find someone else to do it to the best of your ability.
Just to add real quick to that, luck and serendipity is a huge piece of it, always is. But luck and serendipity happen to people who are out there doing stuff, not people who are watching Netflix on their phone. So, full stop. Yeah.
I've talked with quite a few of you, and just people have asked me my career path. I won't go over that, but for those that are specifically thinking industry, and this is, I think, as I've heard, this has been one of the big shifts. I think back to 20 years ago, very few of my colleagues in graduate school were thinking of going to industry. Everybody was thinking of going to academia, and it's just, and now as I talk to a lot of you, it's like, oh, I want to go to industry. I want to go to industry. So here would be my big advice in terms of how do you succeed in that type of an environment. You are your knowledge, your subject matter. I mean, you are the expert in that. But realize, as you get into this environment where you are working with a lot of people, and you are ultimately working for a business, trying to learn as much as you can about other aspects of it. Think about it. You're going to be working with people in sales, marketing, research, people that are engineers possibly, and taking little pieces from each of them to try to understand what they deal with, and then how your piece of it can fit into that, I think can help you to definitely become very, very successful. And as I tell everybody, this is probably one of the biggest things. Be a good communicator. While you have a lot of information in your head, I'm going to be brutally honest with you, 99% of the people you will talk to don't care, or they're not going to understand it. So make sure that you can convey your message simply and just be able to get that across so it can be understood by a wide range of people.
Just to add to Matt's points, as a just tenured professor, I had a sort of a different career pathway, but I share the same thoughts that young students need to make yourself unique. What I meant is there is a fast development of modeling techniques, sensor techniques. Think about how to take advantage of these new things, the AI-related new things, how to use them in your own research. They're just tools. You can use them in any way. And then the AI-related work is usually one of the most interdisciplinary area. So by understanding your, let's say, plant physiology or plant breeding or genetics, then also know some data science, machine learning algorithms, you're going to be in that very specific overlap between the different areas. And then when you are entering the job market looking for a job, doesn't matter if you're going to work in the industry or academia, that very specific interdisciplinary overlap, like the area that you know of, will make you unique and will make you the best candidate among others. I think that will be my, so I can share a little bit of my experience. I was trained as a potato physiologist, and I got my first faculty position as a potato production-based physiologist. And then I realized that the real research I can work is going to be very narrow. I do not have many options to apply funding, and the community I work with is so small. I want to broaden it. How to do that? That is why I started to work with AI 6-7 years ago after I joined UW, came back. And instead of being the traditional agronomist who is doing multi-year of varieties, nitrogen treatments, irrigation treatments, years and years of repeating the same thing, now I'm entering this AI-related area, and I found a lot of exciting opportunities to collaborate with my peers in academia, but also collaborate with our industry partners. So that is going to be my thought.
Great, thank you. We're going to hit another Slido question for. Okay, sorry, because I also want to make sure the ones that people asked earlier asked. For Alex, I have learned that counting the concentric rings in trees can be indicative of the age of the tree. How accurate is that, and does it have any application?
Well, it can get a guy put in jail for 65 years for murder. Yeah, so some species pretty much physiologically have to make a growth ring every year or they die. So it's extremely reliable in those kinds of species, but many woods can skip rings depending on what happens. So it's not entirely reliable. And there's a whole field of dendrochronology where you do cross-dating and line these things up, and that's not really my expertise. But yeah, you can do amazing things with tree rings in terms of figuring out provenance, timing of things. So if you have an established and anchored tree ring record, and then you find another piece of wood from that region of that species, and you measure its rings, you can figure out exactly when it grew. And they have like in the Southwest, like the Laboratory of Tree Ring Research at Tucson, they have chronologies that go back thousands of years. So they can go in and like with Native American habitations, they were able to show that certain timbers were moved unreasonable distances through the desert to go build a house somewhere else, and that this tree would have been cut down in 1403. So yeah, there are lots of applications.
So I wanted to kind of ask broadly about the impact of maybe unusual partnerships, collaborations on your work, and actually I started thinking about this. I'm going to point this towards Nate in terms of protecting endangered species in Hawaii, right? Can you, would you, should you partner with horticulture companies and I don't know if you have Home Depot in Hawaii, but get endangered species out on, for sale to the general public, so they can be growing up things in their backyards. That because seed banks aren't going to be the only answer. I don't know. Maybe that's problematic, but I'm just thinking outside of the box partnerships and how that can impact your work.
Yeah, it definitely comes up a lot. Yeah, it's a little bit of a touchy subject and a complicated issue with Hawaiian flora. And in some cases they do. I think the issue, we see a lot of Brigamia insignis. There's a lot of they have clonal material that Home Depot will sell, and then people will plant a bunch of them, and then you have the exact same clone all over people's yard. We've talked more broadly though about common seed and having more of a native seed supply chain. And I was actually just having this conversation earlier. The biggest issue with that is just actually getting the seed. So we recognize that's an issue, and we want to have this seed supply. Actually, since building this site, I have a contact us page, and it has my office number on it. And 90% of the phone calls I get are from people asking if they can buy seeds from me. But if you take out the endangered flora, we only really have 700 native species, and there's about almost 4,200 species that occur in Hawaii. So it's making those collections, getting those, finding a place to store them. We're not known for our vast amounts of land, but looking into trying to get a warehouse, a facility to facilitate gallon-sized production. And so it has been brought up, and we're working on it. And so at least it's something that continues.
One of the topics that continually came up today was the mountains of data that we are accumulating and how we can process this. One of the questions that we've received is there a way that we can reduce the amount of necessary data collection to still achieve our end result goals?
I think we probably do over-collect in certain situations. As a person who does research in this field, my feeling is that I would prefer to have too much than too little, because you sort of are afraid that you'll miss something if you don't have high enough resolution or frequent enough data acquisition. But at the same time, obviously there are resources that go into acquiring that data and keeping it and processing it. And so I think in applications where we have a pretty good handle on what we're trying to do and what we need to achieve that goal, it's absolutely possible to pare back the amount of data that we're collecting. When we're doing very exploratory work, we err on the side of more rather than less. And so sort of that gathering of more has to come before the gathering of less, at least in my situation.
I worry about not necessarily collecting less data. I worry about paralysis by analysis, that we have so much data that we must prove everything to ourselves, that we miss the real truths in the field that a trained agronomist can identify. I think back to, Alex, what you're talking about of you can look at the wood and and we can build a model and let other people see it. And sometimes I worry that we have so much data, we're missing the true expert in his field just going and doing it. So that's one of my fears about how much data we have.
And another thing, just practically, once you have a mountain of data, then you can do ablation studies. What's the least I can get away with and still have it function? And that's a totally useful, endeavor to do, because if you do that with a couple disparate data sets, you come away with the sense that, oh, I don't actually need 100,000 images. I actually only need N for whatever my N is. So you're working backwards on it's usually quite productive. It saves you a lot of time later on.
Yeah. Downsampling and evaluating the efficacy of your methods can be a really nice method. I was also going to say, I see students sometimes who are really hot to trot on using, say, image analysis to answer a question, or to collect image data or sensor data or whatever to measure something, and sometimes that's very warranted, and sometimes you can just measure it faster and more accurately by hand with a ruler. Like, just get out there, do it, be done with it, don't think about it again. So it is always worth like taking a step back and asking yourself if you're trying to do something a particular way because you feel like you should do it that way, or if it's because it's actually the most efficient way to get something done.
Great. Okay. So a question for Matt. Nutrients are important for plant growth. What about the producer of nutrients? And is there a piece on the pyramid that focuses on microbes in the soil?
Yeah, great question there. One of the things that when this whole process started and we thought about the mountains of data we have and how do we sift through that, it really did come down to this concept around soil health. Now, I don't love the word soil health or the two words together because it doesn't really tell us a whole lot. But, at that time, there had been some tests that came out. Cornell had one. There were a few others. And so we did start to look at that to see from the productivity of the soil, and especially looking at that microbial aspect of it, could we start to take some learnings away from that? And I'll tell you, in the early phases, there wasn't a lot. And I think that, as we continue to delve down that, as we think about soil science, soil testing, all of the different processes that have been worked on in research, I would estimate we probably understand somewhere around 1% to 2% of what actually happens in the soil at this point right now. So it's going to continue to take more of that. Now, at the same time, can we start to look at things that we have a fairly strong belief that they are going to continue to help those microbial populations in the soil? Take cover crops. I think that is something that has shown benefit. The challenge with cover crops right now are that it takes time. It takes continual learning. And, the other interesting part with cover crops, when you look at the amount of acres that they sit on, it's a very, very small percentage. So I think that is going to be something that we will continue to build on. I think the tests will get better. There will get more understanding with that. We just have to I guess, continue to go through the work to really find those processes that help us to really understand what kind of an impact they have.
With that, I don't think we're going to be able to fit in another round of questions. So I want to thank everyone first for showing up and asking great questions. But the big thank, obviously, goes to our speakers today. Thank you very much. This panel was very good. Great organic conversation throughout. I want to thank you. And with that, we're going to close the panel discussion and hand it off to Audrey Morrison for closing remarks.
Are you a walk and talk, or are you?
Awesome. Well, thanks again, everybody. And before I get to thank all of everybody, I just want to say a general thank you so much to everybody in attendance. Whether your involvement today was as an attendee, you brought a poster, you gave a talk, you were a speaker, you traveled from outside universities, you came for the free food. Thank you. We're really grateful for every single person that came today. And as we met weekly leading up till today, and we saw the names popping up that had registered or signed up, it made us very excited to figure out who we'd get to connect with today. So thank you very, very much. So this event is part of the Corteva Symposia series. We are so thankful for Corteva AgriScience's continued support of plant sciences here at UW and promoting this community through our shared passion for plant science research. It is really special to recognize that we have had the honor of being a part of the Symposia series for 14 years now. Thank you again to our other sponsors for this event, Syngenta, PepsiCo, Lima Grain Vegetable Seeds, the Wisconsin Experience Grant, the Associated Students of Madison, Hartung Brothers, Ball Horticultural Company, the Wisconsin Soybean Marketing Board, and Bayer Crop Science. Thank you to Wisconsin Agricultural and Life Sciences Alumni Association for funding this event as well through the Student Impact Grant, and for the UW-Madison College of Ag and Life Sciences for matching their sponsorship. Thank you to Sarah Friedrich from the Botany Department for her incredible graphic designs of our flyer and banner outside. Thank you to our faculty members and our advisors for being so patient and supportive of all of us council members while we worked so hard to plan this event, and it did take a lot of our time. Thank you to Dr. Jason Rauscher for your work in facilitating such a successful Symposia Series network and for your guidance throughout the planning process. And a very big thank you to Morgan Tice, the event manager here for the Discovery Building. Morgan received so many emails from me, several weekly, as we were gearing up towards this event, and she entertained every question and helped make this event happen. And we're also so thankful for John in the back for facilitating our AV help. Thank you to our students from our outside universities for expressing interest to our event, showcasing your work, and making the effort to travel to Madison. Again, we would like to recognize and congratulate Ashley Barstow from North Dakota State University and Julian Cooper from the University of Minnesota. Thank you to graduate student speakers for sharing your research today with us through posters and lightning talks. And a big thank you as well to undergraduate students who were brave enough to also showcase your research through lightning talks or posters. As I gear up to announce the winners, I'll say for the winners, hang tight, and I'll give you directions of how to claim your prize afterwards. But for every student's involvement today, thank you. Even if you did not win, we're so grateful that you signed up and were brave enough to try. So thank you. So I'll take this opportunity to announce the winner of our Lightning Talk competition. You all did a really, really great job. The third place winner for the Lightning Talk competition was Zoe Ryan. The second place winner for the Lightning Talk competition was Julian Cooper. And I am happy to announce that Francisco Campos received the most votes and has won our 2024 Plant Sciences Symposium Lightning Talk competition. Congratulations. Congrats and thanks to everybody that participated. So now we can get into the poster section, beginning with our undergrads. Our third place award goes to Lucia Heiser. Our second place award goes to Jonathan Martin. And finally, our first place award goes to Logan Holland. Congrats, undergraduates, and thanks for being here today. We are really excited this year to have more undergraduates involved in our symposium. And for the graduate student poster competition, third place goes to Zoe Ryan. Second place goes to Jessica Rohde. And we are happy to announce that the first place winner for the graduate student poster competition goes to Darni Babu. But of course, thanks to everybody that brought a poster today. So for all winners, please meet us up here by the stage to receive your prize. For all other participants, thanks again for your participation and bravery in showcasing your research, and we'll be emailing you certificates next week for your participation. And of course, a big thank you to my fellow PSGSC board members, Vice President John Searle, Treasurer Anson Sue, Student Engagement and Outreach Rep Jillian McGee, Agroecology Rep Colin Cooksey, Agronomy Rep Arthur Franco, Botany Rep Joseph Alston, Entomology Rep Fletcher Robbins, Horticulture Representative Erica Kirchhoff, Plant Breeding and Plant Genetics Rep Tony Shue, Plant Pathology Rep Sarah DeSouza, and our soil science representative, Wang Luo. PSGSC is fully run by volunteers. A graduate student's first responsibility is their research, which consumes a lot of time. This group here further elected to add on their volunteer service to PSGSC and other campus and life obligations and responsibilities, which is nothing shy of a full plate. I'm grateful for each of them, and I'm honored to call them colleagues. Graduate students in attendance, if you're interested in serving on the 2025 team, we will be having elections in a few weeks, opening in December. Be sure to check your email for the nomination form, which will be followed by the election ballot. And finally, a big thank you to our speakers today. Nate Kingsley, Dr. Joe Gage, Dr. Alex Wiedenhoeft, Stephen Schwartz, Dr. Matt Clover, and Dr. Wang. Please join me again in thanking them once more for their great talks today. So this concludes today's program. We hope you join us next fall for the 15th Annual Plant Sciences Symposium. Best wishes to everyone who attended, and we look forward to connecting with you again soon. If anyone is interested in continuing conversations, we will be moving across the street over to the set in Union South, where we have 2 bowling lanes reserved, and we'll be getting some apps and some beer. And anyone's welcome to come across and all hang out and be together in one space one last time. Invited speakers and council members, please gather up here by the stage so we can take a group photo. And again, thank you so much for everyone for being here today.